Thank you so much for the introduction, and thanks IPC for this kind invitation. Okay. As we heard today about from very nice studies that psoriasis mainly come back to the previously lesional or resolved skin because of tissue-resident memory T cells. But whether epidermal keratinocytes also play a role in psoriasis local relapse is not clear yet. That's why we mainly focus our research to find out the differences between psoriasis never-lesional and resolved keratinocytes, and to find out transcriptomic differences between never-lesional and resolved epidermis. We performed RNA sequencing on resolved and never-lesional samples, epidermal samples, and since DNA methylation and DNA hydroxymethylation regulate transcriptomic profile, we performed 5-MC and 5-HMC immunofluorescence staining, and we found 5-MC and 5-HMC amounts were overall lower in resolved epidermis than never-lesional epidermis. And loss of 5-hydroxymethylcytosine is also seen in psoriasis lesional skin, and somehow we can see that the disease 5-hydroxymethylcytosine pattern is still remained in psoriasis resolved epidermis. We also found 5-hydroxymethylcytosine weaker intensity was accompanied by decreased T3 mRNA expression level, and T3 mRNA expression level repression, which can catalyze 5-methylcytosine conversion to 5-hydroxymethylcytosine, can explain somehow the loss of 5-hydroxymethylcytosine in psoriatic resolved epidermis. And interestingly, T5-hydroxymethylcytosine pathway perturbation is also seen in psoriatic lesional skin. With our RNA-seq data, we found differentially expressed genes in resolved epidermis compared to never-lesional epidermis, and 102 genes found overlap between lesional and resolved epidermis. Between highly dysregulated genes in resolved epidermis, we found 3 interesting genes which known to be associated with psoriasis pathogenesis, which I will explain a little bit more in the following slide. And the overlap genes were enriched in WNT, TNF, and mTOR signaling pathways. As I mentioned earlier, there were 3 interesting genes known to be associated with psoriasis, highly dysregulated in resolved epidermis. The first one is SAMHD1, which has a crucial role in genome integrity, and it was highly downregulated in psoriasis resolved epidermis. The second one was AKR1B10, which plays a crucial role in vitamin A metabolism, and its overexpression can cause cell hyperproliferation and also higher cell survival. It's worth to mention here that AKR1B10 is not only highly upregulated in resolved epidermis, but also extremely highly upregulated in psoriasis, keloid, atopic dermatitis, and also leprosy lesions. And the last but not the least, GPR15L, which can act as a chemotactic factor to recruit GPR15 positive cells. GPR15 positive cells are mainly memory and also effector B and T cells, and also regulatory T cells, and it's highly upregulated also in psoriasis lesional skin and wound healing, and it's highly upregulated here in our finding in resolved epidermis. Whether the interaction between GPR15L and GPR15 positive cells play any role in psoriasis local relapse is not clear yet, but these 3 interesting genes are very good candidates to be for further studies in the future. And our take-home message is, in addition to tissue-resident memory T cells, we believe that disease-residual epigenetic and transcriptomic profile retained in the resolved epidermal keratinocytes may play a role in psoriasis local relapse. And thank you so much for your attention.
Thanks a lot. Confirming topics we heard this morning. Thanks, Marta. Any questions? So for the sake of time, we'll continue. It's a very brief one.
Oh, it's a really nice talk, and I'm really interested in it. And I wonder that, what do you think if it is the open, the open, sorry, the open chromatin?
Yes.
Yeah, it may the reason that the DNA methylation in fact affect the open degree of chromatin of genes that leading to the innate immune trained innate immune.
Immunity. Yes.
Yeah, immunity, sorry.
Yes, no problem. I understood, and thank you so much for your question. I think you meant that whether the DNA methylation can affect the chromatin condensation and yes, I think so. And regarding our results, we found that 5-MC also was downregulated. So, I mean, the amount was lower in psoriasis-resolved epidermis. So it means that the chromatin is more open than the never-lesional epidermis. Also, 5-hydroxymethylcytosine was lower. So I think it might shows that the chromatin is more open in resolved epidermis than never-lesional epidermis.
So do you think it is possible?
Maybe with ATAC sequencing, we can find more accurate data whether the chromatin is or which part of chromatin is open still in resolved epidermis. But with these findings, we at least we know that the level of 5-MC is lower. So maybe it shows it's open, more open.
Yeah, thank you. And sorry, I have another question. That why didn't you just perform the single cell sequencing instead of that single cell sequencing?
We didn't perform single cell sequencing, but actually that would be great if we can in the future. Yes.
In this way, you can just say that it's keratinocyte.
Yes. Yes. Thank you for your suggestion.
Thank you very much.
Thank you.
Thanks a lot.